focus on regulatory mechanisms that change, in particular when levels of calcium ion in the cytoplasm of the cell change. And again, in us, calcium levels change, and it's a very important regulatory signal in muscle cells. Calcium signals go up in response to contraction. I talk about that in Bio42.
I remember that.
All right, very good. And in your brain, calcium is, where it's at. We think, we remember, all kinds of signals are transmitted by changes in calcium on the inside of the cell in response to some kind of a change on the outside of the cell. So we focus in yeast on understanding how one particular calcium-sensitive enzyme works, and that enzyme is a phosphatase, which means it's going to remove phosphate groups from proteins. So that means I have to talk for a couple minutes about phosphorylation. Again, if you remember from Bio42, we also talked a bit about that. Phosphorylation is really one of the most common ways that protein activity is changed inside the cell. So again, think about your yeast cell. It's growing happily, and then it starts raining, like today. And all of a sudden, conditions are completely different. It wants to be able to respond to things very quickly. So changes in phosphorylation are a way to really quickly change the activity of proteins that already exist inside the cell, as opposed to making a whole new set of proteins, which also happens. But for that, you require transcription to occur and protein synthesis, right? So it's going to be a little bit slower. So there are really fast responses, and then responses that come on a little bit later. But phosphorylation and proteins that add phosphate groups, which are called kinases, and then proteins that remove those phosphate groups, which are called phosphatases, those are key regulatory enzymes. So we study calcineurin, which is, as I said, a phosphatase that's regulated by calcium. And again, I like to draw parallels between what we're doing in yeast and then, why it's important to understand these things and put it in the context of human health. So, for example, calcineurin in yeast is absolutely required for those cells to grow when there's a high concentration of salt in the media. So we can do two things. We can compare the growth of cells that have genes encoding calcineurin that are intact, and then we also have mutant strains that lack those calcineurin genes. So if I showed you those two strains growing under conditions where there's sort of normal levels of salt, you wouldn't be able to see any difference between the growth of those two strains. But if you took those same strains and now tried to grow them on media that had higher salt concentrations, you'd see that the wild type cells adapt and grow fine under those conditions, whereas the mutants lacking calcineurin wouldn't be able to grow at all. So in us, calcineurin turns out to be expressed in every cell in your body, and it's also really critical for different kinds of adaptation to extracellular changes or extracellular signals. But just to highlight a couple things, calcineurin is really important in our immune system. So there's a type of cell called a T cell in your body. Again, Bio42, right?
Yes, everything
It's the best course you're ever going to take, Bio42. Those cells have a job which is specifically to respond to a foreign protein that's in your body, right? Like something like when you have a viral infection. The virus is going to present all kinds of new proteins, and your immune system has the job of recognizing those proteins and then mounting an immune response. And T cells are critical in that. Well, T cells have a protein on their surface that binds to foreign proteins, but then that signal, the fact that the protein is bound on the surface, has to actually cause a change on the inside of the cell, right? Calcineurin. There's a change in calcium, calcineurin gets activated, and that's what allows that particular type of T cell that was able to bind to the foreign protein, causes them to start proliferating. And again, calcineurin has one job. It doesn't know if it's in a T cell or if it's in a brain cell or if it's in a yeast cell. What it is designed to do is be activated in response to calcium, so it can then go dephosphorylate proteins inside the cell. So in us, not only is calcineurin doing important things in our immune system, it's really important in neurons. It's actually 1% of protein in the brain, and really involved in learning and memory, things like that. It's also really important in muscle and in heart cells. It turns out that people that have heart disease, something happens called cardiac hypertrophy, where when the heart isn't pumping effectively, the body actually responds by making that heart grow bigger. It doesn't work, so it's not really a good response, but it's an indication that there's some kind of problem with that heart. And calcineurin is one of the main things that's triggered that then causes that hypertrophic response. So calcineurin is this key protein that's basically sitting there in the cell, and when calcium levels rise, it gets turned on and starts dephosphorylating things. And so back to yeast, because that's what we work on. I was telling you that, for example, whatever calcineurin is doing inside the cell under those conditions, somehow it's actually allowing the cells to grow on these new conditions, right? So one of the main things that we are focusing on is really trying to understand at a detailed level, what are all those proteins inside the cell that calcineurin is dephosphorylating? And what do they do? And how does that event, that dephosphorylation by calcineurin, how does it change the activity of those proteins so that we're now actually allowing the cell to grow in the presence of salt? So we're really trying to map out pathways where at the top of the pathway, I always draw, so I'm not able to do that right here. But at the top of the pathway would be a signal, which for the yeast would be the presence of salt. And then there would be the next event, which would be change in calcium. Then that change in calcium is going to turn on calcineurin, and then the question is, what happens next? What does calcineurin actually do? And as an example, we know there's a transcription factor that calcineurin dephosphorylates. This is again going to relate to Bio-42. And when calcineurin dephosphorylates that transcription factor, it rapidly moves from the cytoplasm, where there's no DNA, right? So it can't be transcribing genes. Once it's dephosphorylated, it moves quickly into the nucleus, where it binds to the promoters of genes and starts modifying gene expression. So that's an example where we can say, here's the calcium signal, calcineurin gets turned on, this transcription factor moves into the nucleus, that starts turning on the expression of new genes, and then we can actually determine what are those new genes and what kind of proteins do they make. And for example, one of the new genes that's activated makes a protein which then gets sent to the cell surface, and it's capable of actually pumping an ion, and that ion is sodium. So when there's lots of sodium on the outside of the cell, one of the things the cell does is to trigger this calcineurin-dependent response, turn on expression of a gene which makes a protein which is present in the plasma membrane and can actually pump sodium ions out of the cell to help respond. So that's one example that we understand very well, but we know that there are lots of other things that calcineurin is doing that we don't understand so well. So that's really what we're trying to do.